The brain is the most complex organ in the human body, with 86 billion neurons, each having thousands of connections. Despite all this complexity, when the brain fails due to aging, it fails in but a few ways. Alzheimer's disease is the most common form of neurodegeneration, accounting for at least 80% of dementia cases and affecting 600,000 to 700,000 people in Canada today. In the Alzheimer's disease brain, the primary molecular hallmark is the accumulation of amyloid-β plaques. The neurons that make up your brain are enclosed in a plasma membrane that is crucial for supporting action potentials and sending important signals. The pathology of amyloid-β is heavily dependent on the lipid bilayer of neurons. First, in that it is the site of production, and second, that it aggregates and binds to the membrane, causing damage. For the last 3 decades, since the identification of amyloid-β as the major constituent of senile plaques, efforts to understand and target amyloid pathology in Alzheimer's disease has been a major focus. Unfortunately, these efforts have not been the silver bullet that early animal studies suggested. Numerous high-profile immunotherapies targeting amyloid have not met primary endpoints in large-scale stage 3 clinical trials. This would indicate that the significant damage to the brain is done prior to the earliest detection currently possible, or that the amyloid pathway itself is not a sufficient target for intervention, and that other factors must be at play. In the first instance, earlier diagnostics would be necessary to identify those at greatest risk of developing Alzheimer's disease, and in the second, more research into other amyloid-related pathways needs to be considered. In a collaboration between Dr. Michael Beasley from the School of Pharmacy and Dr. Zoya Leonenko's group at the Department of Physics and Astronomy, we are using a series of nanotechnology platforms of atomic force microscopy in combination with traditional molecular biology and new lipidomics tools to advance research into amyloid-related pathways involved in Alzheimer's disease. Neuronal membrane cholesterol composition will first be modified, then cells will be treated with amyloid systematically. Under physiological conditions, we will run a battery of tests before and after treatments to study the changes induced in the neuron. First, imaging of the neurons with AFM will be done to characterize their cytoskeletal structure and morphological changes. For instance, amyloid binding to cholesterol domains in a monolipid membrane as shown here. Next, we will probe the mechanical properties of neurons in response to treatments using colloidal bead cantilevers and nanoindentation. Finally, we will use biologically functionalized AFM probes with covalently linked amyloid to study amyloid binding to the neurons and their membranes. These combined studies will shed much needed clarification on the role of the lipid membrane in amyloid toxicity, and we hope will be a useful strategy for diagnostic and drug discovery applications.